Hello everyone, thank you for joining me today. My name is Quintero, and I will be presenting our work of the high-quality genome assembly of Solanum tuberosum group Phureja, specifically the variety Colombia, and its comparative analysis with the public double monoploid genome. This research is a collaborative effort between the National University of Colombia, Francisco José de Caldas District University, and the Brigham Young University. The queer potato, Solanum tuberosum group Phureja, is an essential crop in Colombia, both nutritionally and economically. Colombia produced over 2 million of tons of potato annually, and the queer potato makes up a significant part of this production. However, despite of the importance, the queer potato has been underrepresented in genomic studies. This research aims to fill the gap by providing a high-quality genome assembled by the queer potato and examine its unique genetic characteristics. Our ultimate goal is to leverage this data to inform crop improvement and conservation strategies. The main objective of our study is to assemble a high-quality genome for the Solanum tuberosum group Phureja variety Colombia using PacBio high-five data. Specifically, we set out first an assemble the genome with PacBio high-five technology, and two, compare its structural variation and unique features with the double monoploid genome. Our methodology began with the DNA extraction conducted at the Francisco José de Caldas District University in Colombia. Sequencing was performed at the Brigham Young University in the United States using the PacBio high-five technology. This platform was chosen for its ability to produce highly accurate log reads, which are essentially for assemble complex and repetitive regions of the genome. The data has been processed through our bioinformatics pipeline, where high-five ISM was used for genome assembly, followed by the quality checks and contamination removals. That's what you see in the diagram. In terms of assembly statistics, our genome of Solanum tuberosum group Phureja variety Colombia achieved a significantly higher continuity coverage compared to the DM reference genome. For example, the N50 for the Colombian genome is 82 megabytes per, compared with the 59 megabytes per of the double monoploid, with a total assembly size of the 1.5 gigabytes per. Additionally, chromosomal level analysis revealed that the chromosomes in the Colombian variety are larger, such chromosome 1 is 132 megabytes per compared with the 88 megabytes per in the double monoploid. This difference provides a deeper foundation for future comparative genomic studies. The dot plot analysis shows a high level of synchrony between the genomes, as illustrated by the continuous diagonal line that indicates strong structural conservations. However, we also observe scarred points of the main diagonal, suggesting potential structural variation or rearrangement unique to the Colombian variety. Some chromosomes in the Phureja genome, as seen, are larger, aligning with the assembled statistics, with hinting at additional sequence or duplications. These unique structures may contain adaptive traits relevant to the Colombian agriculture. Using TraitBit, we place the Colombian genome within a phylogenetic context of the Solanaceae family. The analysis continues its close relationship to the other species in the Solanum genus, particularly Solanum tuberosum subspecies indigenus, which is a species cultivated in Colombia. This contextual information provides a comparative framework for further evolutionary and functionally studies with the Solanaceae tribe. In conclusion, we successfully assembled a high-quality genome of Solanum tuberosum group Phureja variety Colombia, with greater continuity and coverage than the double monoploid reference. Comparative analysis showed both structural conservation and unique variations, which may reflect adaptive traits. This genomic research is valuable foundation for future research in breeding and conservation, with potential application in enhancing crop productivity, resilience, and nutritional quality. Future studies can leverage this data to identify the specific adaptive traits and guide molecular breeding efforts. I I would like to extend our gratitude to the International Atomic Energy Agency for their support under the project called 5026. Their funding and resources have been instrumental in advancing the research. Additionally, I thank you and thank all our collaborators and institutes involved in this study. For further details, please visit our website. Thank you everyone for your attention. I will be really happy to take any questions you may have.